professor of the Department of Integrative Biology and Physiology at University of California, Los Angeles. Dr. Paul received his bachelor's degree in biology from Howard University, followed by a PhD in biology at Georgia State that he got at 2003. And then after that, he was awarded a postdoctoral fellowship from Northeastern is my university, Northwestern University in Illinois, at the Center for Sleep and Circadian Biology under the guidance of Dr. Fred Turek. And in 2006, he accepted a faculty position at Morehouse School of Medicine, where he stayed for 10 years there. And after that, he was recruited to UCLA, and he's been there since. Dr. Paul's research interests focus on examining the genetic regulation of sleep, and more importantly, how genetic heterogeneity influences in the ability to recover from sleep loss. His research program currently has two aims. First, to determine if disruptions of biological timing result in sleep-wake disorders, and if so, which specific genes are involved, and to determine if sex differences in the risk and severity of sleep abnormalities are chromosomally driven. So Dr. Paul conducts a forward genetic approach to facilitate gene identification that takes advantages of natural variation occurring in sleep-replete and sleep-deprived mice. This approach applies the most cutting-edge genome mapping, positional cloning, and DNA sequencing techniques to identify the genetic origins of unique sleep phenotypes in transgenic mice models. And finally, these studies are expected to identify novel sleep regulatory genes and lead to the development of new therapeutic targets and improved treatment for sleep disorders. Without further ado, I have Dr. Ketema to give us a wonderful presentation. Dr. Paul, thank you.
Thank you for that generous introduction. Today I'm going to talk to you about the molecular regulation of sleep-wake cycle by circadian clock genes. So since this is not a primarily audience of primarily circadian researchers, I'm going to do the first part of the presentation is going to be kind of a review of what was known in the field about the ability of circadian clock molecules to regulate sleep. And then I'm going to talk about some work that I published a couple of years ago. I know there are a couple of people on the call who've already seen a version of this talk, so forgive me if it's repetition. But as we're recording talks, I've pulled a lot of my more recent data out of them. That being said, I'm going to start out with a model that's well known to both sleep and circadian researchers, and it's the two-process model first proposed by Alexander Borbet in 1982. And what this model suggests is that sleep is regulated primarily by two processes. The first are circadian rhythms that are generated by molecular clocks. These clocks occur in cells throughout the body. And the primary molecular oscillator I'm going to show a cartoon of in the next slide is conserved throughout much of phylogeny, particularly in mammals. And the majority of cells with circadian clocks the clocks are driven by pretty much the same components. There are some other components and a few cell mechanisms, but the majority of the components are very well conserved. And we know a lot of them. We know a lot about rhythms and how they work, relatively speaking. Sleep homeostasis is the other process, and that is sleep homeostasis is not as well understood as circadian rhythms. The most simple way to describe it is responsible for the ability to recover from sleep loss. So if you have a normal sleep-wake cycle and you spend a night awake or you lose sleep during a night, during your subsequent sleep opportunity, you will sleep more deeply and you will sleep for a longer duration. This ability to recover from that sleep loss is very important because it restores a lot of the cognitive abilities and the mental acuity that's lost when you're sleep deprived. We all know that when we're sleep deprived, we can, sometimes it has negative effects on our memory and our ability to focus and concentrate. Well, when you recover that lost sleep, those abilities are restored. And sleep homeostasis not only describes the mechanism that allows you to recover from lost sleep, but it also encompasses those mechanisms that restore the acute changes that happen with sleep loss, and also the chronic changes that happen with sleep loss, like those that are related to comorbidities, comorbid diseases. Here's a cartoon of the basic elements of the circadian clock, and it's a 14-year-old cartoon that I've kept because it's the simplest one to date that I can find, and I enjoy it. I think you guys can see my pointer here. So this is essentially a feedback loop of genetic upregulation, transcription, and translation of several genes that we call clock genes. And these genes, their interaction with one another, their feedback interaction, is primarily responsible for driving rhythmicity in the variety of physiological functions and behaviors, sleep-wake cycle, rhythms of endocrine function, are all driven by these clocks that oscillate close to 24 hours and are, one, able to be entrained or synchronized by the environment, and are also able to time a variety of processes. The genes that are important in this figure are, all of them are actually important, but the ones I'm going to focus on for the sake of this presentation are BMAL1 and clock. These are two positive regulators of the feedback loop. Once they're upregulated, their protein products actually activate PER1 and PER2, CRY1 and CRY2, which are negative regulators of the feedback oscillator. They are converted into protein products. They dimerize, come back into the, from the cytosol back into the nucleus, and have a negative interaction on BMAL1 and clock. Some of the other players in this loop are the RORs, the reverb alpha, casein kinase epsilon and delta. All of these molecules are important. And if you shut off or manipulate the majority of these molecules, you have impacts on the circadian clock. These genes are important not just from a regulatory perspective, but also in a clinical perspective, because there are a number of disorders that are the result of dysfunctioning of this circadian clock. Two of the two sleep disorders that are most prevalent as a result of molecular disruption of circadian clock are advanced sleep phase syndrome and delayed sleep phase syndrome, which the slide in front of you shows. There have been studies that have shown that either polymorphisms or mutations of many of these circadian molecules, PER2, casein kinase 1 delta, PER3, PER2, and casein kinase 1 epsilon, that naturally occurring mutations or polymorphisms, different alleles that exist in families, are responsible for actually advancing or delaying your sleep phase rhythm, where it becomes difficult for you to maintain normal sleep-wake behavior. These are three examples of circadian polymorphisms that have been linked to actual sleep phenotypes that are not associated with timing. So at the top, a group led by Simon Archer and Malcolm von Schantz and Dirk van Dijk found that a polymorphism in the PER3 gene causes an increase in the daily non-REM sleep amount. This is in humans. A group led by Ying-Wei Fu found that the timing gene DEC2, a polymorphism in that gene encodes sleep length in humans. And then a group led by Theo Roenneberg found that a polymorphism in the clock gene is associated with sleep duration in two independent populations. This is important because up until these findings, we kind of were under the impression that these molecules' primary goal or their primary role in regulating sleep was one of timing, of determining when sleep onset and offset would occur, and perhaps some roles in sleep consolidation. This was the first evidence that these timing molecules might actually play a role on regulating sleep duration and other sleep phenotypes that are not necessarily associated with timing. So that's to say that clock gene mutations have a variety of effects on sleep and wakefulness, in which my lab became interested in sometime around 2008, 2009. This is a figure from the Viola et al. Paper that was from Dirk Jan Dijk's group that shows that the PER3 polymorphism causes an increase in non-REM sleep amount in humans. These figures on the left show slow wave activity and alpha activity. Now, both of these are spectral ranges of the EEG. So these are studies using EEG or electroencephalogram in patients. And when you look at EEG, you can measure the energy of any particular bandwidth. If you measure the energy of slower waves that are more prevalent during non-REM sleep, these are called delta waves, and they're primarily within the 0.5 to 4 hertz frequency range. They give you an indication of sleep depth or sleep pressure. And this group found that the polymorphism caused changes. Members of a certain family that expressed the polymorphism had higher intensity sleep during non-REM sleep and higher REM alpha activity, which is also a measure of sleep quality. These graphs on the right show that individuals that had this polymorphism in the PER3 gene, PER stands for period, also showed higher amounts of non-REM sleep during baseline recording and during recovery from sleep deprivation. They also showed lower amounts of REM sleep during baseline and recovery from sleep deprivation. Again, punctuating the point that this gene that's most normally associated with timing and regulating the clock has effects on sleep-wake duration. Then this group led by Yinghui Fu found that the DEC2 gene encoded sleep duration in humans. This is an actogram using accelerometry to show activity patterns across, that's about 10 days. These periods of heavy activity are presumed to be the daytime when we're active. And these periods of quiescence are in the night when we're sleeping. And what they found was the sleep duration was reduced in animals that had this DEC2 genetic mutation. Mouse models and animal models have been very valuable in our understanding of how these circadian genes influence the sleep-wake cycle. This is a study done by Joe Takahashi and Fred Turek's group in 2000, looking at the mutation of the clock gene being one of the canonical genes I showed in the earlier slide. They induced a mutation called the delta 19 mutation, which is a loss of function mutation. It eliminates I'm sorry, it's a change of function mutation. It disrupts clock function. And what they found is the graph on the top shows total amount of wake, non-REM, and REM sleep, and then REM over total sleep time during 24 hours. This middle graph shows during the light phase when animals are asleep more because mice are nocturnal. When animals are asleep more because mice are nocturnal, and then during the dark phase when animals show more wakefulness. And what they found was that the delta 19 mutation in a homozygous mutant animal caused an increase in wake amount, and that additional wake was at the expense of non-REM sleep. So these animals had more wake, less non-REM sleep. This was the first evidence in an animal model. And animal models are valuable because we can do a lot broader range of experiments with animals, obviously. And we can introduce mutations that in the human studies I showed you earlier, those mutations were naturally occurring mutations. The mouse, as you will see throughout my presentation, has been a very beneficial model for us to study sleep in because they are mammals, and although they don't sleep quite like human beings do, their sleep is relatively similar, and they have a lot of the same genes. They have 98%, I think, synteny with the human genome. So we can introduce mutations in many of the same genes and look at their effects on sleep. In this study, they used EEG to measure sleep similarly to the previous study in humans that I showed before. This was a study done by John Weiser in 2002 showing that the double mutation of the cryptochrome gene, the CRY1 and CRY2, has effects on total sleep amount. This double mutation makes the animal arrhythmic. The animal does not show behavioral rhythms in DD, in constant darkness, although they will show behavioral rhythms in a light-dark cycle. And he recorded baseline under a 24-hour light-dark cycle. That's the dark phase, and that's the light phase. He also recorded sleep during 6 hours of sleep deprivation and then subsequent recovery. And these graphs show REM sleep on the top, non-REM sleep in the middle, and then non-REM delta power at the bottom. Now, I showed a graph earlier that showed slow wave activity as a measure of sleep intensity. Delta power is essentially the same thing. What we do is we measure the power of all of the EEG waves between 0.5 to 4 hertz. Again, that's the predominant wave that occurs during non-REM sleep. And what Dr. Weiser found is you can see these gray bars represent areas of significant differences under each graph, and that he found that the double cry mutation, even when animals were rhythmic in LD, he found that there was a significant increase in REM sleep during the dark phase and a decrease during the light phase, and cry 1-2 double knockouts. And he also found differences in REM sleep and non-REM sleep during recovery. Once again, showing that and it's also important to point out that these differences are not the same differences that the clock mutation showed in mice. Both mutations disrupt circadian timing, but they have different effects. He also showed that there's an increase in delta wave or delta activity or slow wave activity throughout the baseline recording period, throughout sleep deprivation, and throughout recovery. So this raises an interesting discovery, and that is that even though both mutations disrupt the clock, they have different effects on sleep, suggesting that perhaps that their ability to regulate sleep is not dependent on the circadian clock, and they may have extra circadian roles in regulating the sleep-wake cycle. This is in DD, where animals are arrhythmic, showing that the rhythmicity that was endowed by the light-dark cycle was not necessary for the genetic mutation to cause differences. This is, again, REM sleep and non-REM sleep and slow-wave activity. Slow-wave activity, as you can see, has the same phenotype in DD as it did in LD, again, showing that the presence of rhythms is not mandatory in order for these mutations to have effects on the sleep-wake cycle. And then Peter Shiramani, who's now at the Medical School of South Carolina, did a study in the PER2 double mouse mutant. This mutation also causes disruption of circadian rhythms. When these mutations disrupt circadian rhythms, what happens is the mice sleep spontaneously on and off throughout the 24-hour period. This mutation is also only arrhythmic in DD, but they have rhythms in LD. And in this figure, Peter did these experiments in LD, and he found that the PER1, PER2 mutation actually reduced REM sleep and slow wave sleep amount during the first half of the active phase, of the arrest phase, which is the light phase. Once again, he showed an increase in wake amount, which is similar to what we saw in the clock mutation, but it occurs at a different time. Again, to punctuate the point that the mutation that has negative effects on the circadian cycle does in fact have effects on non-REM sleep and REM sleep. Slow wave sleep is also non-REM sleep, on non-REM sleep and REM sleep duration. So these studies had already been done at the time or they were kind of in motion at the time we were doing our studies, which I'm going to show you in these presentations. But this got us answering two questions. One is, does the circadian clock have a role in regulating sleep outside of its timing regulatory role? And two, do the individual genes that regulate the circadian clock have sleep regulatory roles? In our study, we chose BMAL1 to look at. We chose BMAL1 primarily because it is the only gene in this molecular clock whose deletion completely obliterates circadian rhythms, both in LD and DD. The other genetic mutations, a single mutation does not have negative effects on the clock, which is why the majority of those studies showed either double mutants, or in the case of the Delta 19, a mutant that was not a knockout. The BMAL1 gene, which is expressed in tissues throughout the body, if you delete the BMAL1 gene, you disrupt circadian rhythmicity. We were also interested in BMAL1 because at the time we thought that its primary expression was in the brain and skeletal muscle tissue. We didn't appreciate the degree to which it's expressed in other tissues. BMAL1 actually stands for brain muscle aren't-like factor, but it's expressed much more greatly in brain and skeletal muscle tissue. than in other tissues. So we chose to look at BMAL1 to see if that gene and disruption of BMAL1 had any effects on sleep phenotypes. At the time, the Fred Turek group had already done studies in BMAL1 knockout mouse, and this figure here shows the results they got from that study. They looked at a wild-type mouse, a BMAL1 heterozygote, and a BMAL1 double knockout. And as you can see, the BMAL1 double knockout is completely arrhythmic in an LD cycle, which is what we would expect, whereas the heterozygote and the wild type are both showing rhythms of total sleep, non-REM sleep, REM sleep, and body temperature throughout the 12-12 light dark cycle. What they also showed was that there was an increase in total sleep time during the dark phase and over 24 hour of recording in the double knockout. And that total sleep time was primarily because of the increase of non-REM sleep, but also an increase of REM sleep contributed to that. And that is the exact opposite of what we see in the clock mutant. Now, clock and BMAL1 are both positive regulators of the circadian clock, and they actually form dimers together in order to regulate the clock function. So we found it interesting, based on Aaron's paper, that the knockout had the exact opposite effect on sleep as the clock delta 19 mutation. However, the BMAL1 knockout mouse is not a healthy mouse, as I will show you in a couple of slides. So at the time that Aaron did this study, he found that the knockout of the BMAL gene increased baseline sleep amount at the expense of wakefulness, increases baseline slow wave activity. Like I said, we measure slow wave activity during non-REM sleep to look at sleep intensity, and it attenuated the ability to recover from sleep loss. But as I said earlier, the BMAL mouse is not a healthy mouse. So we had no idea if any of these phenotypes that we saw were the results of pleiotropic effects of the mutation. And frankly, none of the circadian mutants that I showed you prior to now could we have answered that question, whether the effects that, remember, circadian clocks are expressed throughout the body. And believe it or not, your peripheral interacts with neural structures in order to regulate sleep. So we did not know whether the effects we were seeing on sleep were driven primarily in the brain or driven primarily in the periphery. We assumed they were driven in the brain, but until now, no one had provided really conclusive evidence of that. So we decided to ask two questions. The first, are the influences of BMAL1 on sleep driven by brain-specific mechanisms or by ancillary effects of the mutation in the periphery? And two, are the influences of BMAL1 on sleep related to its role as a clock regulator, or is BMAL1's influence on sleep driven by some extra circadian function? So at the time, I was just starting my lab up at Morehouse School of Medicine, and Joe Takahashi had published a paper where he was able to rescue BMAL1 function specifically in the skeletal muscle and in the brain of knockout mice. That's the figure I'm showing in front of you. He was able to drive an exogenous BMAL1 with a hemagglutinin tag with an active 1 promoter that expressed BMAL1 in the skeletal muscle of BMAL1 knockout mice. So he was able to rescue BMAL1 in the skeletal muscle of knockout mice. And he also developed a brain rescue model on a tetracycline transactivator, which not only allowed us to rescue BMAL1 selectively in the brain while keeping the rest of the mouse as a knockout, it also allowed us to activate and deactivate the mutation using doxycycline. Now, this is important for two reasons. One, because it allowed us to look at specific tissue specificity of BMAL1 expression. And two, this one allowed us to raise the mouse with wild type function in the brain and then shut it off as an adult to look at the effects on sleep. And this provided an added measure of rigor to our experiments, so we can know that the effects we were seeing were actually constitutively driven and not because of any potential developmental effects that occurred in the brain from BMAL1 being knocked out. And we took these BMAL1 models, we implanted them with EEG and EMG recording electrodes, and we put them in a 24-hour light dark cycle. Our first result was pretty surprising. We kind of took for granted that when I should back up. When you rescue BMR1 in the skeletal muscle, the mice remain arrhythmic. They have no circadian rhythms, but many of their pleiotropic phenotypes are rescued. They no longer show arthritic phenotypes that are associated with accelerated aging, and many of the other locomotor phenotypes are restored. So the mice are relatively healthier. They're still arrhythmic. If you rescue BMR1 selectively in the brain, the mice show normal circadian rhythms that are entrainable by their light-dark cycle, but they still are unhealthy. They still show arthritic phenotypes. They still are unable to put weight on their weight-bearing joints, and they're still relatively sick. First finding was when we rescued BMR1 selectively in the brain. We found that brain-rescued mice slept similarly to knockout mice. These graphs show non-REM sleep, REM sleep, and non-REM slow wave activity over 24 hours of EEG, EMG recording. The graph on the left, I'd like to draw your attention to because that's where we saw our significant effects. Along the vertical axis, our total amount of non-REM sleep over 24 hours of recording. And the black bar shows wild type. This is our knockout, and this is our brain rescue. We got the expected phenotype in our knockout. They had dramatically more non-REM sleep than wild types, which we were expecting. We thought that rescuing BMR1 in the brain, because it restores circadian rhythms, would restore non-REM sleep, but it did not. When we rescued BMR1 in the brain, those mice slept like knockouts and slept significantly more than wild types. So rescuing BMR1 in the brain was even though it was sufficient to restore circadian rhythms, was not sufficient to restore wild type sleep phenotypes. However, when we rescued BMR1 in the skeletal muscle, surprisingly, it did rescue sleep phenotypes. We rescued BMR1 in the skeletal muscle. Again, the animals are still arrhythmic. We did not restore any circadian phenotypes. However, the animals slept similarly to wild types and significantly less than the BMR1 knockouts, showing that rescuing BMR1 in skeletal muscle restores sleep phenotypes to those that are in wild types. It was a very surprising result for us, and it took us several times doing the experiment before we were able to convince ourselves that it was real. And till this day, we're still showing that. This graph is to show that that in an LD cycle, the non-REM sleep and REM sleep are indeed arrhythmic in whole body knockouts, rhythmic in wild type mice. Muscle rescued mice, the animals are still showing arrhythmic rhythm, even though we restored the sleep phenotypes. Brain rescue mice, we did restore rhythmicity to the sleep-wake cycle. They have more non-REM sleep in the light phase than they do the dark phase, but we did not rescue the sleep phenotype. So our first and very surprising finding from this was that BMR1 rescuing skeletal muscle restores non-REM sleep amount in knockout mice. However, we were really interested in sleep homeostasis. And the reason this is because we know enough about sleep homeostasis to know that it is indeed regulated by the brain. So at the time, we were kind of assuming that the ability of our muscle rescue to restore sleep was due to some pleiotropic effect in the periphery, that perhaps because the animals weren't feeling as much pain or because of some other effect, that it restores sleep and that it wasn't acting directly on sleep regulatory mechanisms in the brain. But we thought if we saw an effect with sleep homeostasis, which is a brain-driven process, that we would indeed show whether the effects in the periphery were acting on sleep regulatory regions of the brain. So what we did was we did a 24-hour baseline recording, and we did total sleep deprivation during 6 hours of the first half of the light phase and allowed them to recover. And were I in person, I would ask a show of hands, what's the best way to sleep deprive a mouse during in order to get total sleep deprivation. But in this forum, I can't see hands, so I'll just go ahead and give you the answer. And the way we do that is by a technique called gentle handling, where you have to stand in front of the cage and gently tap the cage. And then when the mice get really sleepy, you gently prod them. You do it gently so that you don't activate stress response mechanisms, which are include increases in glucocorticoids and catecholamines. And that's the only way to keep a mouse completely without sleep over 6 hours. Mice are smart, and if you try to introduce an automated sleep deprivation paradigm, they will get what we call microsleep. So those are very brief periods of sleep in which they can dissipate sleep pressure. So if you do this, you either have to spend a lot of money on your workforce, or you have to get really good at seducing undergraduates to come and tap cages. Giving them research credit to do so is a great way to do it if anyone here wants to do a total sleep deprivation. In any case, this is a stereotypical or standard response you get. 6 hours of sleep deprivation is not a extreme amount of sleep deprivation. Here we call it forced wakefulness, FW. And if you do it, you'll get a rebound. That rebound in B6 mice, which is the background of our mutants, normally occurs during the second half of the light phase, but you can see it at any time throughout the recovery period. It usually is expressed as an increase of non-REM and an increase of REM sleep. This is actual data, but I show it here just so you know what to expect from a recovery response to sleep loss. And here you get an increase of non-REM delta power. And it's really important that I point out here that relative delta power, which is the same as slow wave activity, is measured exclusively during non-REM sleep. So it measures sleep pressure, which is kind of the pressure to sleep. A subjective expression of sleep pressure is sleepiness. When you feel sleepy in the afternoons or late at night, that's a subjective expression of your sleep pressure being high. We measure sleep pressure by measuring non-REM sleep when the animals go to sleep, and it's a measure of intensity of sleep, because when your sleep pressure is high, you sleep more intensely. And when we get higher power in our non-REM delta power, as you see here, during the first few hours of the recovery period, the relative delta power is elevated. That is our most reliable measure of a healthy sleep homeostat, that your homeostatic response to sleep is working as it should. So we did that experiment in our brain rescued and muscle rescued mice. And to our surprise, we found out that when we rescued BMO1 in the brain, the recovery response was similar to knockouts, different from wild types. When we rescued BMO1 in the skeletal muscle, we got a recovery response that was similar to wild types and higher than in knockouts. Again, showing that rescuing BMO1 in skeletal muscle had a restorative effect on non-REM sleep and non-REM slow wave activity. These graphs on the right show spectral curves showing you the power across the EEG frequency range from 0 to 30 hertz. As I said earlier, during non-REM, this is during non-REM sleep. And as I said earlier, during non-REM sleep, you get the most of your power in the lower waveforms between 0.5 to 4 hertz, which is highlighted here in yellow. And as you can see, the robustness of the power was as high in muscle rescues as in wild types. Brain rescued mice had delta power, which was similar to knockout mice. This was a very exciting result for us, because this was our first indication that the manipulation in skeletal muscle was indeed acting on sleep regulatory mechanisms in the brain. A slow wave activity measures neural responses in the brain. Which shows us again that the ability of rescuing BMO1 in the skeletal muscle is not necessarily a pleiotropic effect, that the skeletal muscle is some kind of way signaling the brain, as indicated by the slow wave activity, which is similar to wild types. So we found that BMO1 rescuing skeletal muscle restores sleep phenotypes in knockout mice, and that it also alters the ability to recover from sleep loss. It restores the ability to recover from sleep loss, showing that the rescuing skeletal muscle, again, is having direct effects on sleep regulatory mechanisms in the brain. So we knew that rescuing BMO1 in the skeletal muscle was sufficient to restore sleep, but was BMO1 in the skeletal muscle necessary for sleep? Well, in order to do that, we got a mouse model from, and the studies I showed you earlier were done by Dr. Chris Allen at Morehouse School of Medicine. Was when we did those studies. The studies I'm going to show you right now is done by Dr. Allison Brager, who is currently in the Army as a program officer and as a sleep scientist. Here we got a mouse model from Karen Esser, which selectively knocks out BMO1 in the skeletal muscle by, again, fusing the ACTA1, human alpha, skeletal alpha actin 1 to a Cre-regulated and tamoxifen sensitive estrogen receptor, which when you give the animal tamoxifen, it selectively knocks out BMO1 in the skeletal muscle. And as our control, we use the Flox mutant BMO1 with tamoxifen. And this, again, in these mice, animals have stable circadian rhythms because BMO1 is being expressed in the brain. But the knockout of skeletal muscle, which is conditional, we activate it with tamoxifen, does have effects on skeletal muscle tissue, knocks out BMO1 in skeletal muscle. And when we knocked out BMO1 selectively in the skeletal muscle, we were able to increase non-REM slow wave sleep to a level that we would normally see in knockout mice. REM sleep, we saw no effect, and we did not see any effect in our Flox mutant controls, showing that not only was BMO1 in the skeletal muscle sufficient for normal sleep phenotypes in the brain, but also necessary, but knocking it out recapitulated what we saw in the wild-type mouse. Again, this was done by Alison Brager. We also looked at non-REM slow wave activity after sleep deprivation. Again, non-REM slow wave activity is a measure of sleep intensity. First, we looked at total amount of non-REM sleep gained over sleep loss after sleep deprivation, and we found out that the recovery response was impaired in animals in which BMO1 was selectively knocked out in the skeletal muscle. That's something that we saw in the whole body knockout. And we saw the slow wave activity was actually elevated in animals that have the selective knockout of BMO1. And this was during recovery from sleep loss. So I showed that selective knockout of BMO1 in skeletal muscle increases non-REM sleep amount. Selective knockout of BMO1 in skeletal muscle reduces recovery from sleep loss and increases slow wave activity. And that BMO1 expression in skeletal muscle is necessary for proper functioning of sleep regulatory processes in the brain. Okay, all right, so far we've shown that BMO1 expression in skeletal muscle is both necessary and sufficient for normal sleep phenotypes, and that they are necessary for the recovery response from sleep deprivation, suggesting that this BMO in skeletal muscle is acting directly on brain-driven sleep processes. But we still don't know if what we're seeing is the result of, is clock dependent, is circadian dependent, is dependent on the presence or absence of actual rhythms. In order to do that, we looked at BMO in the other direction. Instead of knocking it out, which actually impairs the circadian. And I should also point out that when we knock it out in skeletal muscle, even though the animal has circadian rhythms in the brain and is behaviorally rhythmic, knockout of BMO1 in the skeletal muscle actually knocks out the clock in the skeletal muscle. So while the mouse may appear to have rhythms, the skeletal muscle tissue do not have endogenous rhythms. So in order to test whether the effect in skeletal muscle was dependent on a clock in the skeletal muscle, we overexpressed the BMO1 in the skeletal muscle. And we went back to the models provided to us by Joe Takahashi. This time we expressed both of these transgenes on a wild type background. When you express them on a knockout background, you restore sleep phenotypes. When you express them on the wild type background, you overexpress when you express them on the wild type background, you restore BMO1. I'm sorry. When you express them when you express them on a knockout background, you restore BMO1. When you express them on a wild type background, you overexpress BMO1. So here we use the same transgenes. We overexpressed BMO1 in the skeletal muscle and overexpressed BMO1 selectively in the brain. And when we did that, we didn't find any effects on non-REM or REM sleep duration. When we overexpressed BMO1 in the skeletal muscle, when we look at non-REM, REM sleep amount and relative slow wave activity, and when we overexpressed in the brain, looking at the same variables, we did not see any effects on non-REM or REM sleep amount. As you can recall, knocking out BMO1 increases sleep. We expected overexpressing it would potentially decrease sleep. It didn't in the brain or the muscle. However, we did get this result, which we thought was peculiar when we did the experiment. We overexpressed BMO1 in the skeletal muscle, and we saw that consistently animals that had BMO1 in the skeletal muscle had basal slow wave activity levels that were reduced. Why is that important? As I said, non-REM slow wave activity is a measure of sleep pressure. So if it's reduced under basal conditions, when the animal has not been sleep deprived, it suggests that the mouse has a lower sleep pressure. The mouse is just not as sleepy. It suggests the mouse is potentially more vigilant and less likely to be susceptible to sleepiness. We got really interested in this phenotype. So in order to test it, we took our muscle overexpression, and the muscle overexpression mice, again, are healthy mice and they have healthy circadian rhythms. So if you overexpress BMR1 in the brain or the skeletal muscle, you don't have any negative effects on circadian rhythms. The rhythms are robust. You do have some small effects on entrainment if you overexpress it in the brain, but nothing major. So what we did is we took these muscle overexpression mice and we kind of turned up the volume on sleep deprivation. We gave them 24 hours of sleep restriction. This graph on the top left-hand shows a 72-hour protocol, 24 hours of baseline recording, 12 light, 12 dark, 24 hours of sleep restriction, 12 light, 12 dark, and 24 hours of recovery. Now, what is sleep restriction? Sleep restriction is automated sleep loss, putting an animal in a slowly rotating wheel and letting the wheel turn very slowly to keep the mouse in locomotor activity. As I said before, mice will find ways to sleep. As you can see, these wild-type mice, which are in the black circles with white insides, are getting sleep, and their ability to sleep increases as the day goes on. They sleep in these micro sleeps, and it's really cool when you watch them. Actually, they run up to one side of the wheel and sleep as the wheel turns. And then when the wheel gets to a point, and it turns very slowly, and when it gets to the point that they can't stay rested anymore, they run back to the other side and go back to sleep, which is why we call it sleep restriction instead of total sleep deprivation. We did that, and then we allowed the mice to recover for the subsequent 72 to 24 hours. And our first finding, when we looked at the data, we noticed that the mice that overexpressed BMR1 and skeletal muscles looked like they showed less microsleeps during sleep restriction than the wild-type mice. And when we actually quantified that data over the 24 hours of sleep loss, we did show that indeed mice that overexpressed BMR1 and skeletal muscle did have less sleep than wild types, which suggests to us that perhaps indeed they are having a lower basal sleep pressure, and they were just easier to keep awake than the wild types. And when you look at recovery, it doesn't look like there is a difference in recovery amounts. However, as you can see, these mice lost less sleep than their wild type counterparts. And when you look at non-REM sleep gained over non-REM sleep lost, then it's striking. You can see that the mice that overexpressed BMR1 and skeletal muscle only recovered half the sleep as their wild type counterparts, again suggesting that these mice may indeed be resistant to sleep loss. Then we did something we normally don't do. This graph on the bottom, where we looked at slow wave activity during wake. Now, normally, under normal sleep deprivation conditions, you normally see changes in slow wave activity during non-REM sleep. You don't see a lot during wake. You do get slow waves intruding into your wake EEG, but normally after 6 hours of sleep deprivation, that's not very instructive. It's not significant. After 24 hours of sleep restriction, however, you can get measurable intrusions of non-REM slow waves into your waking EEG. And when we did that, that was our most striking results. We saw that the ability to incorporate slow waves into the waking EEG was much more enhanced in the animals that overexpressed BMR1 and skeletal muscle. Well, what does that mean? That suggests to us that since these animals were having more slow wave intrusions into their wake, that they were indeed resistant to sleep loss, and they were able to stay awake despite the fact that their sleep homeostat in the brain was telling them that their sleep pressure was building and that they needed to get to sleep. They still were able to maintain wakefulness, which does suggest that they are more vigilant and are indeed resistance to sleep loss. And here we show non-REM relative delta power or slow wave activity across the 72 hours of recording, and throughout baseline, sleep restriction, and recovery, we see that what we saw in our original finding is that mice that overexpress BMR1 and skeletal muscle have lower slow wave activity, lower delta power, suggesting that they may indeed be resistant to sleep loss. So in conclusion, to the sleep data, we've shown that BMR1 rescue and skeletal muscle restore sleep phenotypes in knockout mice. Selective knockout of BMR1 and skeletal muscle reduces recovery from sleep loss and increases slow wave activity. BMR1 overexpression in skeletal muscle reduces recovery sleep loss, reduces slow wave activity, and appears to make mice resistant to sleep loss. And BMR1 expression in skeletal muscle is necessary for proper functioning of sleep regulatory processes in the brain. Because my time is short, I'm going to jump past the next slide and show you some of the phenotypes that we saw in skeletal muscle. When you overexpress BMR1, BMR1 is a circadian gene, and we don't know it to be related to structure of skeletal muscle. But we did find out when we overexpressed BMR1 in the skeletal muscle that we did get an increase in the mean cross-sectional area of skeletal muscle fibers. We did not get a significant increase in muscle myofibers, showing that the overexpression of BMR1 in the skeletal muscle does indeed have effects on muscle structure. We also, I'm going to go past that one, saw effects in sleep regulatory regions of the brain. This was work done by Dr. Alison Brager, showing that if you overexpress BMR1 in the skeletal muscle, in the basal forebrain, the lateral habenula, pedunculopontine tegmentum, and the lateral dorsal tegmentum, these are all sleep regulatory areas. CFOS regulates, measures neuronal activity. We looked during control and during sleep deprivation, and we saw that animals that overexpress BMR1 in the skeletal muscle did show more neuronal activation in sleep regulatory areas of the brain. Once again, suggesting that that overexpressing BMR1 in skeletal muscle is having effects on sleep regulatory areas in the brain. And with that, I will close with my acknowledgements. Most of the experiments done in today's talk were done by Dr. Chris Aylen, the Morehouse School of Medicine, Alison Brager in the U.S. Army. These are individuals that assisted in these experiments and the data collection. And most of these experiments were done at the Circadian Rhythms and Sleep Disorders program when I was still in Morehouse School of Medicine. Thank you, and I will open the floor to questions.
Thank you, Dr. Paul, for that wonderful presentation. We have a couple of questions, but before that, I just wanted to ask everyone if you could type in your email if you haven't done so thus far, so we can send you our evaluation form later about how good are we doing with these presentations, and seeing if we're gonna be able to continue them in the semester, perhaps, if we have a lot of people thinking that this is worth doing it again. So I really appreciate if you could just put your email at the chat, and that will be quite helpful. Thank you. Now, we have a question, Ketema. One is, it says, when you recovered BMAL1 in the brain, was it recovered in all cells, neuronal and glia?
It was no. It was driven by secretagranin promoter, which is neuron specific. So it was, so no, we did not, to our knowledge, actually, and I don't know if Joe's gone back and specifically labeled neuroglia. To our knowledge, it was restored primarily in neurons and enriched in the suprachiasmatic nucleus, which is the location of the master circadian clock in the brain. So no, and that's a great question, because obviously the role of neuroglia in sleep regulatory mechanisms is becoming a hot topic these days. Marcos Frank at Washington State and a few others are doing work in this area and finding out that neuroglia does in fact have some sleep regulatory effects. But no, in this model, the BMR1 was not restored in neuroglia, not to my knowledge.
Okay. Then there's another question, says, are there any differences in locomotor activity in the BMR muscle knockout mice?
The BMR muscle knockout mice have severe, severe reductions in locomotor activity. So if you knock out BMR1 selectively in skeletal muscle, you reduce locomotor activity. If you rescue BMR1 in a whole body knockout selectively in skeletal muscle, you restore most of the locomotor activity, but they're still not as active as wild types. You overexpress BMR1, we're not seeing any changes in locomotor activity, except, interestingly enough, in LD during light-dark transitions. Animals that overexpress BMR1 are more active than wild types. We're in the process of writing a paper showing that. And we also saw that in DD, for some reason, mice that overexpress BMR1 have slight increases in behavioral activity. And this is in beam break, looking at in a beam break field. So BMR1 does appear to be important for locomotor activity. Until recently, most of those were attributed to accelerated aging and BMR knockouts, accelerated aging phenotype, because most of the effects on locomotor activity look arthritic in nature. But we are interested in if any of those reductions in locomotor activity are the result of effect of muscle mutations on neural processes. So another great question.
Okay. And then there is another question is, what is your hypothesis of how these changes in muscle affect the brain? And then
Man, that's the $1 million question. So we had delayed putting out this data for a long time because we got kind of exhausted of looking for it. So we put it out, and I've been here at UCLA for about 3 years, and we're still looking. So we don't know the signal between the brain and muscle, but we are getting evidence that BDNF in the brain may be involved. We were also interested in chirenin, a factor called chirenin, and we have done some experiments looking at that and PGC1 alpha, but we haven't seen much in regards to PGC1 alpha. When it comes to sleep, we are doing behavioral experiments. These are all the stuff I took out of this presentation, incidentally. We are doing a behavioral expression that shows a relationship between, so PGC1 alpha is impaired and is altered in animals that overexpress BMAL1. PGC1 alpha is a muscle protein. And we are finding that we are doing behavioral expressions with memory, and we are finding some correlations between PGC1 alpha expression in muscle mice and memory retention in a novel objects maze. And we're currently writing those results up now. We're kind of in the same spot we were when we did these. We keep doing more and more experiments, and we have to try to convince ourselves when we're ready to actually publish. So, but in answer to your question, no, we don't know what the signal is yet. We don't know through what signal the muscle is acting on the brain.
Okay, and then also it's the other question is, what gene or protein changes occur in muscle? Any ideas?
So we did do a proteomic screen of muscle tissue, and we saw a variety of changes. Now that's a difficult one, right? Because BMAL1's a timing gene, and it acts on what are called clock control genes, which do a variety of things in muscle. Most notably, we've seen effects on energy metabolism. So we saw a lot of changes in energy metabolic genes. We saw changes in genes that encode for cyclic AMP levels. We saw changes in a lot of mitochondrial genes, and we saw changes in POMC, interestingly enough, which is a precursor for glucocorticoids. But we don't know very much what those mean. This was a proteomic screen, and the only genes we've actually done muscle assays with are PGC1 alpha, chirenin, and BDNF. So, but we do know that there's BMAL1 has a very large role in energy metabolism in muscle tissue. Garrett Fitzgerald's lab at Penn and someone else's lab at Penn, who's I'm blanking on, have already published data showing that BMAL1 has mutations have effects on redox reactions in mitochondria. Okay. So in answer to your question, energy metabolic genes seem to be the most, obviously circadian genes are also altered. So we saw effects on circadian timing genes too.
Okay. Then another question, we still have time for a couple more. Can you do parabiosis experiments to see if there is a signal from the muscle carried by the blood?
Can we? Yes. Have we proposed those? Yes. Have we gotten funded for those yet? No. So those kinds of parabiosis experiments are, as you probably well know, I'm guessing that's why you asked the question, have been kind of the bread and blood for research in the sleep homeostasis. One of the reasons we know that a homeostat exists is because we've done so many tissue and replacement and fluid replacement experiments. When I say we, I mean the field, showing that if you take tissues or fluid from a sleeping mouse and implant them into a mouse that hasn't been sleep deprived, that you can get that mouse to be sleepy. We would love to do some of those in our muscle experiment, in our muscle knockouts, and we are proposing those for a grant we're writing right now, but we have not had an opportunity to do those yet. Okay.
And then, does skeletal muscle activity, high or low locomotion, affect wild-type expression level of the BMAL1?
The skeletal muscle activity express wild-type expression of the BMAL1. No, we have not seen where we are currently doing experiments where we are actually working the animals out. Putting them on, the animal version of treadmills, making them exerting, because we want to find out if the muscle mutation is dependent on muscle exertion. BMAL1 expression is not dependent on muscle mutation. But the interesting part about that question is that this paper got a lot of press, which I'm happy about. It still gets press because neurobiologists and neurologists are shocked. Muscle physiologists were not surprised at all. Alison Brager, who did much of the work on this, has a lot of interest in muscle interactions with the brain. She published a book on it recently. And so she kind of did a pass when we were writing this. We gave it to a few exercise physiologists to take a look at it. They weren't surprised at all. There are myriad reports of muscle exertion having effects on sleep pressure. Actually, one of the big fights that was in the field and actually still is in the field is muscle fatigue related to sleep homeostasis. Believe it or not, there's still a large group in the sleep field that don't believe that it is. We're getting evidence from our lab, and others have gotten evidence, and a lot of them exercise physiologists have evidence also that muscle fatigue does contribute to feelings of sleepiness and to sleep pressure. So that's also one of the $1 million questions, and we're currently doing experiments in our mouse models to look at those.
And finally, any gender differences? Are you looking at males and females, or?
Yes, so that's one of my areas of expertise. I've published a lot of papers in the area of sex differences, and I'm currently renewing a grant looking at that. But in this case, no, that we saw the exact same effects in all of our models in males and females, and that's rare. Actually, we're thinking that might hopefully give us some clues as to how this is acting. The majority of sleep phenotypes show some even mild sex differences, but in BMAL knockouts, we're not the only group that have looked at sleep in these mice. We're seeing that we're getting the same effects in males and females. So which kind of would maybe suggest that the circadian clock does have a role. And I should also point out that most of the phenotypes we saw were not circadian dependent, but there are some phenotypes that are circadian dependent that we did not show in this paper. So we're still trying to tease out the precise role that the clock plays on the ability of BMR1 and other molecular components to regulate sleep, but it does look like that it is doing so equally in males and females. So this is one of the few phenotypes of sleep which is not showing a sex difference.
Wonderful. That's very interesting as well. Well, I think we are ready to finish our presentation. And thank you, Dr. Paul, for being here today and giving us such a wonderful talk on circadian rhythms and the molecular underpinnings of it. And please join us next week when we have our very own Dr. Jose Garcia Raraz giving his talk on regeneration of the nervous system. So
I will be there, and I'm hoping that you guys have some sort of program in the future. You can invite some of your speakers to come back to give an in-person talk, because I would love to visit.
Of course, to do that. We will. And thank you, Ketema, for being with us today. And thank you all for participating in our seminar. And I hope to see you next week. So
thank you for having me.
Bye.
Bye. Take care.
Thank you. Bye, Ketema. Bye.
Bye, Carmen.